Colorectal cancer, as with all other cancers, is triggered by an abnormal growth of cells that can invade and spread to different parts of the body. In this tumor type, abnormal growth originates in the colon or the rectum. Colorectal cancer, known as CRC, is the third most commonly occurring cancer in men and the second in women. According to the World Cancer Research Fund, there were over 1.8 million new cases diagnosed in 2018. Nowadays, especially in the current era of precision medicine in oncology, new approaches for the treatment of cancer are rapidly emerging, including the development of therapies targeting molecular alterations or signatures. It is thanks to such progress that we are seeing improved outcomes for patients with CRC. Motricolor, spurred by Horizon 2020's European Union funding for research and innovation, is a European project incorporating 10 different institutions spanning 4 European countries, working in partnership. 8 of them are leading research institutions linked to hospitals, and the other 2 are a small, medium-sized enterprise specialized in molecular diagnostics and a contract research organization. This consortium aims to match specific treatment strategies to colorectal cancer patients based on their particular molecular alterations. But how does it work, and how do researchers develop these novel treatment strategies? Motricolor is conducting 3 different early-phase pioneering clinical trials to establish the anti-tumor activity of novel therapies for patients with metastatic or advanced colorectal cancer. The first step of the project is to stratify patients according to their gene expression profiles. Agendia, a leading molecular diagnostics SME, identifies those patients presenting specific molecular alterations that may be sensitive to the therapies under study. All patients are recruited from the Motricolor clinical sites. If the molecular profile of a patient matches any of the Motricolor signatures, this patient is included in one of the studies. Each patient receives the treatment locally, meaning that all clinical trials are being conducted simultaneously across the 7 recruiting sites. In parallel, translational research is also being carried out. Through the 3 clinical trials, Motricolor aims to identify indicators of response to treatment, known as biomarkers, mechanisms of secondary resistance, and the emergence of resistant clones by tracking and closely monitoring circulating tumor DNA. By studying the molecules present in blood, we seek to establish why a particular therapy may cease to be effective. In addition, we are focused on advancing immunotherapy-based approaches against cancer. Our ambitions center on 3 different goals. First, to develop or repurpose 3 novel therapeutic strategies for CRC patients with advanced disease. Second, to perform pioneering research that will lead to the development of the first companion diagnostic test in CRC. And thirdly, to potentiate the efficacy of these therapeutic strategies.